Hello, everyone. My name is Shireen. I'm one of the founders of the Lyme Care Resource Center. Welcome to our evening. We join in this community to receive support in helping us navigate the journey of Lyme disease, either in ourselves or in someone we care about. Here we focus on mind, body, and spirit, and the interdependence between these. The nature of community is also one of interdependence. So another goal of our gathering together is to be reminded that we are not alone. Each time we begin, I share one of the principles upon which this Lyme Center was founded, and that's the connection between mind, body, and spirit. Our sense that things are separate comes in large part from the physical boundaries we see in the dimensions of time and space. Our bodies, for example, have distinct shapes, height, width, depth, surrounded by space, which separates them from one another. Certainly, the concepts of mind and spirit exist beyond the typical boundaries we encounter. Those who have explored the ideas of connectedness recognize that it includes energetic forces, potentials, auras, and even higher selves. One tool in the realm of connectedness, which has been shown to be of incredible benefit to many aspects of our well-being, is mindfulness. Research has shown that mindfulness affects different aspects of the neural circuitry and thereby changes our physiology. Mindfulness fosters connection on many levels. But what is it about mindfulness and being in the now, bringing our awareness to the present moment, that helps us to make connections, which in turn contribute so greatly to our well-being? We know that mindfulness improves cases of depression and anxiety, past and future thinking, in which our minds are absorbed elsewhere. Our minds are often wrangling with past events and future potentials. Contrasting this with our bodies, our cells are always in the present moment. For our bodies, like nature and the universe, it is never not now. There is a pace at which our physiology is most comfortable, but whether our cells are in sympathetic fight, flight, or whether they are in parasympathetic rest and digest, they are always operating in the present moment, doing what they must to maintain or resume homeostasis in the here and now. And yet, if our minds are going incredible distances at lightning speeds for indefinite periods of time, then it seems that mind and body are often operating as two incongruent energetic potentials. So when we practice mindfulness, or even just a momentary meditation, perhaps we create some link, some connection. Bringing our minds into the present moment, the here and now, do we create some energetic congruence with our cells, our physiology, which is always existing in the present? And is it then that this congruence between mind and body is one of the links that fosters the benefits of mindfulness, or at very least, that facilitates the relaxation response? Using the breath, that bridge between conscious and unconscious, to focus our minds on that to which our bodies already adhere, one can imagine that we create some synergy that is otherwise absent. Regardless of our circumstances and surroundings, when we turn down the noise around us, it seems that we foster the peaceful connection to that which is within us, always here and always now. In its purest form, mindfulness connects us with the self, and this connection in turn fosters deeper connection with the world around us, just as it is in any present moment. This evening, we have a very special guest who is going to speak about connections between the brain and the body. Earlier this year, I was looking into non-pharmaceutical interventions for GI distress when I came across a beautiful paper on vagal stimulation and its effect on GI motility. I saw a contact number listed, and so of course I dialed. What I anticipated was reaching an admin or an assistant who might guide me to more resources. And what I got was a very kind and gracious lead researcher, Dr. Roberta Sclocco, who spent a good deal of time discussing her research with me. And after all that, she did not even hesitate when I asked if she would be willing to share her time and knowledge for the benefit of this community. Gratitude for someone like this is a given, but words do not adequately convey the feeling of humanity and grace we experience as the beneficiaries of such interactions. Thank you, Dr. Sclocco, and now I'll pass the screen to our director, Gina Costa, for her welcome and more informative introduction of our guest for the evening.
Hello, welcome everyone. Thank you for signing on tonight. Shireen, thank you for your introduction. You shared some really amazing talking points that really help us to each reflect on the ways we can intentionally choose to walk our paths, focusing on being present, being positive, and being purposeful. Those are just perfect focal points of the night. And as we are always focused on healing and providing education through our live streams, it really feeds into our goal of connecting with each and every one of you that sign on. We care so much about you, and we don't want you to go through this journey alone. We all know what that feels like, and we have our ups and downs. And when we say that we don't want you to go through this alone, we mean that we want to support you in taking control of your health. We want to support you in making small positive changes that lift you and encourage you and motivate you. We want to encourage you to follow your intuition and seek out a caring community that's going to guide your healing. We want to give you strategies, and we want to explore them together as it relates to restorative sleep, nourishing nutrition, good self-care. We want to really support and cultivate this community. And it's challenging right now. We're doing it from our homes, but we have 5 years of a lot of effort and a lot of amazing keynote speakers that take the time to give back to the community, to educate us and to guide us in our journey, to know that there are moments when we feel uncertain and we feel like we don't have a lot of choices with our illness or with our health challenges. But when we take in the education, it broadens our horizons and it helps us envision more. It helps us sometimes go deeper in our faith, to connect more deeply with other people. Sometimes it helps us to think about our strong reasons for living and our desire to see above the storm, which is a really significant parts of healing and wellness and optimal health, all of the things that we're striving towards. And also to put meaning into our stories and our life experiences. So we're thrilled to have all of you tonight, and I want to take a moment to talk a little bit about our keynote speaker. Tonight we're going to listen to a more scientific talk, and a lot of times we will explore mind, body, spirit resources. Tonight we're going to be listening to a talk by Dr. Roberta Sclocco, and we're going to focus on how strong and capable our bodies are. We're going to really sit back and listen and take in the presentation. We're taping this presentation to be able to provide it to you and to others in our group that weren't able to sign on tonight. So feel free to just receive the information. If time allows, we're going to have some time at the end for questions. So feel free to write in throughout the presentation any questions that you might have that can be addressed at the end. We are so honored to have such a distinguished scholar and presenter this evening. Dr. Sclocco is a woman of many, many talents. From achieving her PhD to her extensive research on the gut-brain axis and the vagus nerve, working in the laboratory, and extending even further into her faculty role in the classroom as an instructor at the Martinos Center. She wears many hats and she offers many gifts to share. We are so grateful for your time tonight in creating a presentation and for your give back and sharing your research and expertise. I wanted to share a quote as it relates to time. The greatest gift you can give someone is your time, because when you give your time, you're giving a portion of your life that you will never get back. So this ties in nicely to our earlier message of being present, that being present is truly a gift, and that's why it's called the present. But your presentation tonight, Dr. Sclocco, and your work has the opportunity to positively influence each and every one of our healing journeys. So I'm going to turn the presentation over to you, and we just want to extend our warmest welcome. Thank you.
Thank you, Gina, for this really great introduction, and thank you everyone for tuning in for my talk. And as Gina said, I'm an instructor at the Martinos Center for Biomedical Imaging at the Mass General Hospital in Boston. And tonight I will talk about my research on investigating and modulating the gut-brain axis in humans. So please come with me on a journey from the ear to the stomach, and let's see where we get to. So I'd like to start with a little bit of an anecdote. More than 10 years ago, when I was looking for a project to work for my master's, I went to talk with this senior professor, and he asked me, do you want to work on the brain or the heart? And I remember thinking, why should I choose only one? But those were my options, so I started working on the brain. But since then, for all my research career up until today, I always try to integrate the brain investigation with other peripheral organs that, of course, are participating in how the brain works and how everything talks to each other. So this is some background of my research interests. I can also share with you that I work in a neuroimaging lab, and where we mainly focus on brain responses to aversive symptomatology, and specifically chronic pain. However, I've always had a personal interest in brain-body interactions and have looked for opportunities to explore this topic throughout my career. Over the last few years, with my mentors and collaborators, I started focusing on the vagus nerve, since this provides a physical connection between the brain and numerous organs. This cranial nerve carries preganglionic fibers involved in the autonomic regulation of several organs, including the heart and parts of the gastrointestinal tract, or GI tract. And it is responsible for tasks such as heart rate, GI peristalsis, sweating, and other functions. Therefore, accessing the vagus nerve and somehow modulating its activity could constitute a powerful tool for many clinical applications. In particular, tonight I'd like to talk about the stomach, which is innervated by both branches of the vagus nerve that regulate its so-called accommodation, that is the ability to expand in response to food, and peristalsis, meaning the contraction waves that move the food downward to the small intestine. The main body of the stomach can be divided into a top portion called fundus, which expands when we eat, and a bottom portion called antrum, where the contraction become vigorous to push the bolus out through the pylorus and into the duodenum. Of course, the stomach is not an isolated organ, such as the brain is somehow, but it is in close proximity with other abdominal organs, such as the liver, the intestinal tract, the spleen, etc. In a coronal view of the abdomen, like the one on the right, the stomach is marked with the number 3. The stomach has an intrinsic electrical rhythm, pretty much like the heart, and this rhythm is generated by the interstitial cells of Cajal, which are located in the stomach wall. Vagal sensory neurons are also in this region, and they send ascending sensory information up to the brain. Additionally, the vagus nerve conducts descending modulatory signals from the brain to the stomach. This bidirectional vagal connection is likely important for altered gastric function or sensations in so-called disorders of gut-brain interaction, such as functional dyspepsia or gastroparesis. The visceral information carried by the vagus nerve are then processed by a set of cerebral regions involved in what is called interoception. A recent review updated the definition as the processes by which an organism senses, interprets, integrates, and regulates signals from within itself. The neural circuitry in the brain that support interoception are shown here, notably the anterior cingulate cortex, the insula, prefrontal regions, the somatosensory cortices, several subcortical regions, and a few key brainstem nuclei. Our work is focused on the nucleus tractus solitari, which is a major integration and relay node for visceral sensations carried by the vagus nerve. We are interested in understanding how other cortical regions are functionally connected with the NTS, and how these connections could reflect altered visceral sensation processing in disorders of gut-brain interaction. Therefore, modulating the activity of the vagus nerve implies interacting with the central circuitry as well as with end organs, such as the stomach. But how can we interact with the vagus nerve, and how do we measure the effects of such intervention? An established treatment targeting the vagus nerve exists and is used for resistant epilepsy or major depression. Traditionally, VNS, or vagus nerve stimulation, requires the implantation of a neurostimulating device connected to an electrode located along the cervical branch of the vagus nerve. VNS treatment is approved for drug refractory epilepsy and major depression, as I said, and some clinical trials have tested its use in stroke and cardiovascular applications. But despite the reported beneficial effects of VNS, its invasiveness and complications have limited the conduction of clinical trials evaluating its effect on autonomic modulation for other indications than heart failure. Therefore, the development and optimization of an alternative non-invasive techniques for vagal modulation is of critical importance. A possible non-invasive option is given by the auricular branch of the vagus nerve, or ABVN, which is known from cadaver studies to innervate the human auricle, and most consistently a specific portion called the cymba concha. From there, electrical impulses are able to stimulate the vagus nerve transcutaneously, hence the term transcutaneous auricular vagus nerve stimulation, or TVNS. I'm sorry, but there will be a number of acronyms throughout my talk. So the stimulation of the ABVN is primarily afferent, meaning going up to the brain. And a number of studies have combined this neurostimulation paradigm with functional magnetic resonance imaging, or fMRI, in order to evaluate the brain response to this stimulation. Even though TAVNS has been linked with activation of the lower portion of the brainstem, called the medulla, some technical challenges, and more importantly, a wide range of possible stimulation parameters and paradigms, make it more difficult to combine and summarize the results. In our work, we attempt to optimize TAVNS parameters using brainstem responses from fMRI as outcomes. So as a starting point, we need to know where exactly to look within the brainstem. The vagus nerve includes axons which converge onto two main sensory nuclei. The nucleus tractus solitari, or NTS, which receives visceral and taste afferent information, and the spinal trigeminal nucleus, which receives sensory information from the outer ear, the dura of the posterior cranial fossa, and the mucosa of the larynx. As for the efferent side, so going down from the brain to the periphery, the two main motor nuclei are the nucleus ambiguus, which mainly sends efferents to the heart, and the dorsal nucleus of the vagus, which sends parasympathetic output to the viscera. All these four nuclei are located in close proximity in the medullary portion of the brainstem. So we have established that we need to interrogate the brainstem. At this point, let's consider this structure for a minute. The brainstem connects the forebrain to the spinal cord and acts as a relay and processing station between these parts of the central nervous system. However, it has some anatomical and physiological characteristics that present challenges to neuroimaging analysis. A significant challenge in using fMRI to assess the response to TAVNS in target nuclei is posed by the structure of interest itself. The brainstem has a small volume relative to the size of the brain. And in addition to the nuclei involved in autonomic regulation, which are highlighted in the red box in this illustration, oops, I apologize. Yep. So in addition to these autonomic nuclei, the brainstem is involved in the modulation of many other functions and hosts key source nuclei for several neurotransmitters, for example, the Raphi nuclei regulating serotonin release. The reduced size and close proximity of these nuclei, as well as the location of the brainstem, which makes this structure particularly susceptible to physiological noise, calls for specialized imaging methods. So for example, let's consider a nucleus that is called periaqueductal gray, and it's located in the midbrain, so the upper portion of the brainstem. If we use a standard fMRI voxel that is 3 millimeters in size, it would be covered by roughly 4 voxels. Something like this. However, this nucleus is involved in a variety of functions with functionally distinct subportions that receive inputs and project to different structures. So with our spatial resolution, it would be quite difficult to interrogate a specific subsections of this nuclei. A third and final challenge is posed by the physiological noise. The brainstem is surrounded by spinal fluids and blood vessels, and therefore more prone to physiological noise, which can corrupt the fMRI signal, especially in the dorsal aspect, where many nuclei are located. Therefore, particular care is needed in the analysis stage of brainstem fMRI data, with specific steps that can be performed in order to reduce the influence of physiological noise. So a possible solution is given by ultra-high field imaging, which provides enhanced spatiotemporal resolution and signal-to-noise ratio, thus allowing for smaller voxels. In this example, the difference can be appreciated for the functional MRI data, where subsunction nigra and red nuclei are distinctly visible in the 7 Tesla data, as opposed to the 3 Tesla. So basically, by increasing the magnetic field, we can reduce the voxel size and see more detail in our images. An additional challenge comes from physiology. So physiology tells us that vagus nerve activity, and hence the response in the NTS nucleus, are synchronous with respiration. NTS is a key node in respiratory sinus arrhythmia, that is the mechanism through which the heart rate increases during inhalation and decreases during exhalation due to regulation of the efferent vagal tone via NTS and nucleus ambiguus. We also know that NTS receives sensory afferents from aortic baroreceptors and lung stretch receptors during inhalation, concurrently with top-down influence from the ventral respiratory group in the medulla, resulting in NTS inhibition during inhalation and facilitation during exhalation. So long story short, NTS is facilitated, meaning its activity is it's more receptive to interaction during the exhalation phase of respiration. So all these factors have to be considered when using fMRI to measure NTS response, so to take into account its physiological oscillations. However, this physiological mechanism can also constitute an advantage in our case. We can then introduce the respiratory phase as a stimulation parameter and hypothesize that given NTS activity being facilitated during exhalation, if we deliver the stimulation during this phase, we can increase NTS activation compared to delivering the stimulation during inhalation. This also gives us the advantage of avoiding continuous stimulation, which could lead to possible habituation of NTS and brain responses in general. We call this paradigm respiratory gated auricular vagal efferent stimulation, which I will refer to as RAVENS in the next slides. In the picture, you can see a diagram of the stimulation setup where the respiration is measured from the subject and used to gauge stimulation in real time. So imagine that this brainstem nucleus is listening closely to the stimulation when you breathe out. And this is what we're taking advantage of with our paradigm. In our first study, we combined ultra-high field fMRI with brainstem-specific data collection and analysis methods to compare brainstem responses to exhalation and inhalation-gated ravens, which I will call E-ravens and I-ravens. We enrolled 16 healthy subjects and used customized MRI-compatible electrodes to deliver stimulation to the Simba concha of the left ear. Subjects underwent separate 8 minutes fMRI scans, during which they experienced either E-ravens or I-ravens. Additional scans included an active control, where the electrode were placed on the left earlobe, which is innervated by another auricular nerve that is not the vagus nerve, and a zero current scan to control for respiratory influence on NTS response. The order of the 4 conditions was randomized across subjects with a similar number of stimulation events, interstimulus interval, stimulation current, and perceived intensity. fMRI data were collected with a partial brain coverage centered on the brainstem. When looking at target nuclei response, E-ravens showed robust activation of ipsilateral NTS, significantly higher compared to I-ravens. As for the active control, so the earlobe stimulation, the response was more consistent with ipsilateral spinal trigeminal nucleus compared to E-ravens. This is a reminder of the importance of carefully choosing controls in whatever electrical stimulation study, especially when considering them as inactive. We also compared the fMRI response to different conditions in 2 serotonergic source nuclei, the dorsal and median raphe, and in a bilateral noradrenergic source nucleus, which is the locus ceruleus. We found significantly greater response during E-ravens in both dorsal and median raphe, as well as in contralateral locus ceruleus. Finally, we used electrocardiographic data collected during the scan to estimate time-varying heart rate variability indices, and looked at the evoked response of the high-frequency spectral component known to be related to cardiovagal modulation. We found that E-ravens enhances parasympathetic output as measured by this index, but this is not found during I-ravens. At this point, we decided to optimize a second stimulation parameter, and we looked at the effect of stimulation frequency on brainstem response. We enrolled 30 healthy controls subjects for this study, and this time subjects underwent 4 E-ravens active scans, each at a different frequency, plus the zero current stimulation scan to correct for respiration effects. The frequencies considered in the study were 2, 10, 25, and 100 Hertz, and were delivered in random order across subjects. The stimulus amplitude, that is the current, was calibrated in order to match the sensation across frequencies and subjects. Therefore, lower current values were used for higher frequencies, as shown in the upper left graph. In this study, we also evaluated whether E-ravens had any effects on respiratory depth or frequency, and we did not find any differences compared to the zero current condition, which is good because we don't want to be modulating the respiration itself. It should be naturally modulated by the person. When looking at target nuclei response to the 4 stimulation frequencies, we found that 100 Hertz evoked the highest engagement in ipsilateral medulla, with a cluster encompassing NTS, spinal trigeminal nucleus, and dorsal motor nucleus of the vagus. The second highest response was found for 2 Hertz stimulation, suggesting a nonlinear dose response curve. A subsequent source nuclei analysis also confirmed consistently greater activations in response to 100 Hertz E-ravens, with significant responses in dorsal raphe and ipsilateral locus ceruleus for 2 Hertz as well. Interestingly, a positive correlation was found between activations in these 2 nuclei at 2 and 100 Hertz, meaning that subjects with a higher response at 100 Hertz also exhibited a higher response at 2 Hz. So now that we have chosen 2 parameters that have shown to enhance the response of target nuclei in the brainstem, we can explore a possible application of ravens. So while our lab has shown acute effects of ravens in chronic pain populations and in major depression, in this talk, I will focus on GI applications, specifically for modulation of gastric function. The vagus nerve is known to play an important role in both sensory and motor aspects of gastric physiology, modulating gastric accommodation, that, as I said before, is the ability of the stomach to expand in response to food ingestion, and gastric motility, that is the ability to generate contractions for gastric emptying. Therefore, modulating vagal efferents with ravens may allow to affect gastric function when this is impaired, such as in gastroparesis or functional dyspepsia. Moreover, our frequency modulation study showed that ravens at 100 Hz is able to engage the dorsal motor nucleus of the vagus, which is the main motor nucleus sending vagal efferents to the stomach. So the question now is, how can we evaluate the gastric response to ravens? We know how to look at the brain, but how do we look at the stomach? So in order to answer this question, we need to know what gastric outcomes we want to measure and how. To evaluate gastric accommodation, emptying, and motility simultaneously, we need a test that provides volumetric assessments continuously over time. One possible option is given once again by MRI, magnetic resonance imaging, that has been proposed as a reliable, non-invasive method for detailed evaluation of gastric emptying and motility without the disadvantage of ionizing radiation and the use of intragastric catheters. An important advantage of using MRI is given by its ability to directly measure gastric volumes and contents. Here, for example, you can appreciate how the gastric content is precisely quantifiable from the MRI images, while a breath test on the same subjects overestimates or underestimates gastric emptying, since it's influenced by absorption rather than volume. Furthermore, MRI is completely non-invasive. In the case of accommodation, a method commonly used is the barostat, where an inflatable balloon needs to be positioned in the gastric fundus and inflated. This is not necessary when using MRI. Finally, MRI can provide information on multiple aspects of gastric function at the same time, without the need for separate tests. A recent review underlined the potential of MRI in clinical GI testing and suggested possible future developments exploiting technological improvements driven by the advance of use of cine MRI or dynamic MRI in the cardiac field. In fact, the main advantages provided by MRI have been so far counterbalanced by as many drawbacks, thus limiting its widespread use. Most abdominal MRI sequences require breath hold in order to avoid image corruption by respiratory motion. Thus, volumetric images covering the entire stomach are usually limited to a single time point. For motility assessments, where images over time are needed, the field of view is generally limited to a single slice, thus losing the volumetric information. Finally, gadolinium is often used as a contrast agent, with contraindication in vulnerable populations such as renal insufficiency, pediatrics population, and pregnant women. To overcome this limitation, we have proposed a free-breathing, 4-dimensional MRI sequence using a food-based contrast that will be used to assess the effects of ravens on gastric function. So we have a study that is still ongoing, and in our study, each subject that is enrolled was asked to fast overnight prior to each MRI visit. And then they received either a sham with zero current or active ravens for the entire duration of the visit. At the beginning of the session, subjects consumed a fixed dose of a pineapple-based semi-solid, high-caloric contrast meal. Following the food ingestion, they were placed in a 3T MRI scanner and continuous abdominal volumetric images were collected for 5 minutes at 3 post-meal time points, namely after 15, 65, and 80 minutes following food ingestion. The cute pineapple that I'm showing here is because the meal-based contrast is based on pineapple, which is very bright in the MRI images due to high manganese content. So the MRI sequence used did not require the subject to hold their breath and provides full coverage of the stomach every 7 seconds. A mosaic movie of unprocessed abdominal images is shown for a representative subject. So following data collection, the gastric volume is segmented within the images via a semi-automated procedure. Briefly, the manual segmentation of the first volume is then propagated on subsequent time points using a nonlinear volumetric warping. Our first paper came out just a few months ago, and we demonstrated to be able to measure gastric emptying, separating meal and air content in the stomach, and also look at gastric motility by identifying the propagation of peristaltic waves along the gastric walls and extracting parameters such as peristaltic frequency, antral occlusion, and velocity of wave propagation. So which gastrointestinal population do we want to look into now that we have this technique? We thought about functional dyspepsia patients. So functional dyspepsia is a common gastrointestinal disorder of the upper GI tract associated with a variety of symptoms, such as abdominal pain and bloating. But it lacks identifiable organic abnormalities. Altered motility patterns in functional dyspepsia occur in the stomach, pylorus, and small intestine during digestive or interdigestive periods. And up to 30% of symptoms are associated with migrating motor complexes-like activity, thus suggesting a link between gastric motility and sensory disturbances. However, gastric functional impairments have proven to be challenging to assess using conventional abdominal imaging, which typically evaluates anatomy at a single time point and relies on a variety of modalities with varying degrees of invasiveness, as we've seen before. So we enrolled functional dyspepsia patients according to the Rome-4 criteria. And these patients were screened by the GI department at Mass General and were required to have undergone a gastric empty scintigraphy to rule out gastroparesis and an upper GI endoscopy to rule out other GI disorders. We also enrolled healthy controls that had no GI issues. The measure I'll be focusing on today is the peristaltic propagation velocity of antral contraction waves. To obtain this measure, cross-sectional area is computed at each slice of the antrum for every volume. The volumetric profile then shows the peristaltic waves of contraction that move downward the antrum. By tracking these waves, we get a measure of how fast the contraction propagates down the antrum. When taking the average velocity across the 3 scans, functional dyspepsia patients exhibit lower peristaltic propagation velocity, even when corrected for differences in ingested meal volume. If we look at the individual time points, we see that this group difference is significant at the first and second scan. Additionally, while the velocity remains stable in the functional dyspepsia patients, there is a significant increase and then a subsequent decrease in velocity for healthy controls. So now we've identified a motility parameter that shows differences between healthy controls and patients. We hypothesized that it might be related to differences in visceral sensation processing. But in this experiment, we also collect brain data in between gastric scans. So since we're interested in visceral sensation processing, we explored the functional connectivity, so the synchronicity of the NTS in the brain imaging data. After pre-processing the data to remove sources of noise, we use the time series of the NTS region as a regressor for every voxel in the brain to generate a 3D correlation map for each scan. So basically, we have a map where each voxel represents the degree of synchronicity between this nucleus in the brainstem and the specific region in the brain. So we then compared the two groups, the healthy controls and the patients, to look at the specific regions that show higher or lower NTS connectivity, so similarity between groups. So we identified several regions that show increased NTS connectivity in functional dyspepsia patients compared to healthy controls. Specifically, the anterior cingulate, pre-supplemental motor area, ventrolateral prefrontal cortices, and the anterior insula. Several of these regions, especially the insula, have been previously implicated in interoception. Now that we've identified differences in NTS connectivity between groups, we wanted to see if the connectivity between NTS and any of these regions was linked to the underlying altered gastric motility. And we indeed found significant negative correlations between NTS connectivity to the left ventrolateral prefrontal cortex and the pre-supplementary motor area and peristaltic propagation velocity in the entire sample. The correlation was also significant in only the healthy control group. The direction of these correlations imply that subjects with greater propagation velocity, so faster gastric peristalsis, have lower connectivity between the NTS and these regions. We then evaluated the effect of Ravens on this parameter, so on the antral peristalsis velocity within the FT group. And we found that peristaltic velocity was increased during Ravens compared to sham, especially at 65 and 80 minutes post-meal. Additionally, we looked at gastric emptying over time in both the sham and active visit in FT patients. And we found that total and meal volumes were significantly larger at T0, so immediately after the meal ingestion, even though subjects consumed a comparable amount of food in the sham and in the active visit. This finding might suggest increased accommodation, that is, the stomach is able to expand more easily in response to food during active Ravens. Moreover, gastric meal emptying measured at 65 and 80 minutes post-meal were significantly higher during active Ravens compared to sham, meaning the stomach is more efficient in emptying during Ravens. And we did not find any significant effect in the control group. So up to this point, in the first part of the talk, we have shown that brainstem neuroimaging using fMRI allows to optimize Ravens' target engagement. We have then described a comprehensive tool based on MRI for integrated non-invasive brain-gut assessments of Ravens' effects for GI applications. Finally, we have proposed an application of Ravens in gastrointestinal patient population and showed promising results of this intervention in modulating gastric function in FT patients. But to put my talk into perspective and to offer some relevance to patients suffering from post-treatment Lyme disease, I'd like to show a recent publication comparing symptom rates and severity between post-treatment Lyme disease patients and controls. As you can see, GI symptoms are not even listed, with the exception perhaps of nausea at the very bottom of the list. Many high-scoring symptoms, however, are related to pain, such as joint pain, muscle pain, neck pain, low back pain, and headache. So as I mentioned at the beginning of my talk, I am part of a neuroimaging lab that specializes in pain neuroimaging. Over the years, the acute effects of Ravens have been investigated on chronic pain populations, such as pelvic pain patients and patients with migraine. Furthermore, we have an ongoing study led by my mentor, Dr. Napadow, which is investigating the effects of Ravens combined with mindfulness meditation as a treatment regimen for migraine patients. This is our first longitudinal study that we're running, and where devices are given to patients to use at home. And migraine patients are evaluated before and after the treatment. Finally, a collaborator, Spalding, and this is something that I knew about yesterday, Spalding Rehabilitation Hospital in Boston, Dr. Ximei Wang, is initiating a study specifically enrolling Lyme patients and using ravens as a possible treatment for chronic pain. The study is not yet recruiting, but if anyone here is interested and perhaps not too far from the Boston area, please feel free to reach out to Ximei or to me, and I will put you in contact for any inquiries. Before ending my talk, I would like to thank my mentor, Vitaly Napadow, and all my collaborators and lab members, as well as funding sources. And of course, thank you for your invitation and for your attention tonight. I'm open to any questions.
Hi, I'm going to jump back on with you at this point. Thank you so much. That was amazing. And it's just incredible to hear the extensive nature of your research. So I'm going to just ask our group to take a moment. If anyone has questions and concerns, I have a couple coming in, but please take a moment to write in for questions or comments in the Q&A right now. And while we're waiting for additional participation, I can start with some of the questions that we have. So the first question, Dr. Sclocco, is auricular stimulation somehow related to acupuncture?
So this is a good question. It is. It is somehow related, meaning that acupuncture traditionally has some points in the ear. I'm not an expert, but my mentor, Dr. Napadow, is also a licensed acupuncturist. And I think that is how he developed his first interest in the Ravens protocol. So it is somehow related. The main difference is that there's no skin penetration. The electrodes are just placed in contact with the skin. However, there is a device which is FDA approved for children, which is percutaneous. So meaning it has some very small needles that break the skin. And it's placed very similarly to the setup that I showed.
Thank you so much. Okay, we have a couple more questions coming in. The next one is, Dr. Sclocco, can you please describe in more detail your study at Spalding Rehab? Who is doing the study at Spalding? Could you please spell the name? Is this at the Dean Center? How do patients connect to the study?
So I wish I could, but I can show again the email of the principal investigator. The study has not started yet. I think they're going through IRB approval and all the study setup. And I don't know of any other details, but please feel free to email me or Ximei and ask about inclusion-exclusion criteria or when the recruitment starts.
And the other thing is, I could also ask for people that are signed on tonight, specific to tonight, if you're interested, you could also, if it's easier, to write into the Lyme Care Resource Center, and then I can connect directly with Dr. Sclocco to help with this. I had another comment come in about the phone number again for the other person on this study. But again, I think we could just address that through, you put the slide back up, but I think we could address that through connecting through me or through you.
Okay.
Okay, so the next question, thank you, is why does it take so long before something like Ravens is available on the market?
Yeah, so myself and my mentor and other lab members, we get many emails even from patients that reach out to us asking if something like the Ravens device is available on the market, where they can buy it, if their doctor can prescribe them something similar. And tonight, I hope I gave you like an idea of the work that is behind the development of any medical device, of anything in general in research, but medical devices specifically, because there are many layers of complexity. And so the studies that I went through tonight span a period of time going from, I think, 2015 to now. And that's just my portion of the work, which had already started years before me. So from the in-house garage looking prototype that we used in our study to a commercial device, which is also FDA cleared or approved because it's a medical device, there is a lot of time and effort.
Thank you. I believe we are through the questions at this point, unless there's any more that are coming in at this time. I don't know, Dr. Sclocco, if you have any additional thoughts or comments that you'd like to share at this time.
No, just thank you for listening and thank you for inviting me. I hope this was at least a little bit interesting for you, and please feel free to reach out if you have questions.
Well, I do have one more that just came in, and so then the next question is, what kinds of endpoints are you going to be measuring in the Lyme disease patients?
Again, I'm not sure. I think what they're proposing is based on a number of questionnaires. So it's mostly self-assessment. And there might be also some blood samples collection, but I don't know the details. I'm sorry.
That's okay. And I know it's always a challenge. We have different speakers of expertise, and sometimes it is all about Lyme disease or tick-borne illness or chronic health challenges. And sometimes it's more about research in the field with what is evolving and growing as it relates to health and disease. So I know that those are hard questions, but we do have some comments that are coming in through the Q&A. One of our participants said, "Tremendous work, great visuals. Thank you."
Thank you.
Nice to share. And then I think another one just popped up. Can you elaborate more about chronic pain and imagery?
So yes. So over the years, our lab has run a number of studies looking at mostly brain plasticity in chronic pain populations, meaning how the brain, not only the structure, so like the real shape of the organ, but the function of the brain. So how different brain regions talk to each other and work together. So how these are modified in people that experience pain over time continuously. And we have found some characteristics that are specific to chronic pain populations. And then we have attempted some intervention that are usually minimally invasive to try and normalize this abnormalities in brain function. So that is basically our work. We have worked with carpal tunnel syndrome, fibromyalgia, migraine, pelvic pain, low back pain, a number of chronic pain populations. I don't know if this answered the question for you. Feel free to follow up.
Yeah, thank you. And if there's any follow-ups to that, please write in. We had another comment that came in saying, "Thank you so much for your work. It's very interesting."
Thank you.
And we have another question. Do you have a time frame for your vagal nerve device?
So we are, meaning my mentor is already collaborating with a company who has developed this first generation device, which is the one that we're using in our migraine plus mindfulness study. So there is a portable version. It's not FDA cleared or approved, so that's why it cannot be sold. As a timeline, I don't know, because it depends on, I guess, the company submitting an FDA request for approval for a specific indication. So it will depend on the indication as well.
Thank you, Dr. Sclocco. I think we don't have any more questions that have come in. So I just want to thank you very much for your time and your presentation tonight. And we will definitely be sending out the replay to our amazing group. And at this point, I'm going to turn it over to Phil Kenny, our other co-founder of the Lyme Care Resource Center, for some information that he'd like to share and some closing remarks. So thank you again, Dr. Sclocco.
Thank you.
This time, I'm going to turn it over to Phil Kenny. Thank you.
Thanks, Gina. And Gina, thank you for putting this together tonight. We definitely are stronger together, folks. Thank you all for participating in our fundraising efforts and celebrating our 5th year anniversary. Together, we raised over $18,000. And a special thanks to our anonymous matching donors who brought it up to that number, and everybody that supports our mission and give back. So we'd like to visit our website, www.lymecare.org. There you can join our monthly emails for distributions of resources and encouraged message. On our website, you'll find replays, recorded replays of our webinars, very good stuff, and view our collection of inspirational quotes. You can print a copy of our wellness tips brochure. On there, there's also the checklist, and use that on a daily basis to make positive changes in your life. I know I have to go back to it myself to keep me on track. Thank you all for joining us tonight. Thank you all. Be at peace. Good night.